this lecture, we're going to look at an anabolic pathway called gluconeogenesis, which is an essential pathway that allows your liver to supply the rest of the body with glucose. There are a couple different substrates that can be used in order to make glucose in the liver, and we'll see where these molecules come into the pathway, and you'll see that because it is an anabolic pathway, we are going to have to input some energy, and that we can't just simply use the reverse of glycolysis, which breaks down glucose. As always, regulation is going to be very important, and we'll look at how gluconeogenesis and glycolysis are reciprocally regulated so that they're not both on at the same time in a liver cell. Just a few supplemental readings to help with your learning. So just back to my cartoon summary of the pathways, we've seen how glucose is broken down to pyruvate, and that can then be used in the mitochondria to eventually produce ATP. Oxidative phosphorylation is the production of ATP and is dependent on redox reactions and the presence of oxygen. If you don't have oxygen, all of these pathways are going to back up and be inhibited. The pyruvate that was made can't be converted to acetyl-CoA, and there's instead a side reaction that occurs producing a waste molecule called lactate. We'll see that it, along with glycerol and amino acids, can be used to make glucose in gluconeogenesis, and even though GNG and glycolysis are represented as separate pathways, they actually share some of the same chemical reactions. And these are going to be the ones that have delta Gs close to 0 and are reversible, so totally dependent on the concentration of substrates and products. As I just mentioned, when you don't have enough oxygen, complex 4 in the electron transport chain can't pass its electrons onto 1/2 molecular oxygen, so the electron transport chain backs up, and all the pathways in the mitochondria are going to slow down. This results in what's called anaerobic metabolism, where glycolysis is the only way we can make ATP. The pyruvates that were made at the end of glycolysis will instead get reduced to form a lactate molecule. And there's an enzyme called lactate dehydrogenase, and since this is an oxidoreductase, the NADH will be oxidized to NAD+. You may have heard about lactic acid buildup when you're running and not supplying enough oxygen to your muscles, but based on what you now know about pKa values and biochemistry, the molecule that's actually made is the conjugate-based lactate. Now, reaction is really important because it'll regenerate the NAD+ that glycolysis needs at step 6. So this will allow glycolysis to still run and make 2 ATPs per glucose molecule, but it will no longer be making NADH in the cytoplasm, which really doesn't matter anyways since oxidative phosphorylation isn't working in the mitochondria. So pyruvate has many fates. It'll be converted to lactate under low oxygen, but it can also be converted to ethanol, and this is known as anaerobic fermentation, which occurs in yeast and helps in the brewing process. Last week, we saw how the pyruvate dehydrogenase complex converts it to acetyl-CoA for ATP production, and it can also be used to make oxaloacetate for the citric acid cycle to run in order to efficiently make lots of ATP. This reaction is also going to be important for the generation of glucose in gluconeogenesis, which we're going to look at in this lecture. We can also use pyruvate to make fats after it's converted to acetyl-CoA, and if you change this carbonyl on pyruvate to an amine, I hope you can see how alanine can be made. But you could also make pyruvate from alanine, breaking down amino acids for energy. So let's look at how the liver can make glucose in gluconeogenesis, or GNG for short. Gluconeogenesis is really important because your brain can't use fats for energy. When you're sleeping or fasting, your blood glucose is going to drop, and your liver can first break down its glycogen to share this storage form of glucose with the rest of the body. But your liver is only so big. Eventually, you will run out of glycogen, and you need another way of making glucose. GNG will take place primarily in your liver, but your kidneys can produce about 10% of the glucose, but really it's your liver that's the supplier for the rest of the body. That lactate that we just mentioned can be used, but there are other non-carbohydrate precursors such as glycerol from fat breakdown and amino acids like alanine that can enter into this pathway. These molecules can travel throughout the bloodstream and get picked up by your liver to use them and convert them to glucose. So remember, glycolysis is on the left, where we were phosphorylating glucose and then breaking it down into 2 triose phosphates. So whenever we're making glucose in the reverse pathway, we do need to consider how many carbons are coming in. And you would need 2 starting molecules for gluconeogenesis. So both glycerol and lactate or pyruvate have 3 carbons in them, so you would need 2 of each of these molecules. The Cori cycle describes this mechanism of using a waste product to make a useful molecule. In your muscle cells, under low oxygen conditions, or really any other cell that's undergoing anaerobic metabolism, lactate will be produced. We're just really considering muscle cells since they are so abundant and need a lot of energy when you're exercising. Your red blood cells actually don't have any mitochondria, so they are always producing lactate. And this lactate is found in the bloodstream, so it can get picked up by your liver for use in gluconeogenesis. That same reaction that we saw, catalyzed by lactate dehydrogenase, is going to work in the reverse, where it's going to use an NAD+ to remake pyruvate and an NADH. And then that pyruvate will get converted to oxaloacetate, either for the citric acid cycle or gluconeogenesis. Amino acids can also get converted to pyruvate or other citric acid cycle intermediates, or we can use glycerol to make glucose. That glucose can then leave the liver and get picked up by other cells for glycolysis and energy production, so it could be used by muscle, brain, or red blood cells. Gluconeogenesis simply isn't the reverse of glycolysis. Remember, when we talked about the thermodynamics of glycolysis, it was favorable in the forward direction, and a ton of energy was released. If we were to just reverse all 10 steps to make glucose, we would need to put in 84 kilojoules per mole of energy to drive it in the opposite direction. Gluconeogenesis is an anabolic pathway where we're going to use smaller molecules to form a 6-carbon glucose, so there will be a few steps requiring energy, but we're also going to bypass those irreversible steps of glycolysis with different enzymes. So in glycolysis, steps 1, 3, and 10 all had very negative delta Gs. So instead of just trying to reverse them, there's actually going to be 4 different enzymes that will catalyze reactions to help make gluconeogenesis favorable as written. So instead of pyruvate kinase, there's going to be a 2-step reaction, and we'll look at it on the next slide, producing oxaloacetate in the mitochondria and then exporting it and making PEP, which then goes through the reverse reactions we saw in glycolysis. And remember, stage 2 of glycolysis happened 2 times, so we would need 2 molecules coming into gluconeogenesis to make our triose phosphates, which then combine to form fructose 1,6-bisphosphate. These 2 new enzymes in pink are going to help in gluconeogenesis to remove the phosphates and eventually produce glucose for the rest of the body. So let's start by looking at how gluconeogenesis can make PEP, and remember that was the molecule with the very large phosphoryl transfer potential. The reverse of step 10 in glycolysis requires 2 enzymes that are going to use some energy to produce phosphoenol pyruvate. The first step is adding on a carbon dioxide molecule, and bicarbonate is actually going to be the molecule that will donate this CO2. The enzyme that catalyzes this reaction is called pyruvate carboxylase, so we're carboxylating pyruvate, and it is a ligase, so it's going to use the energy in ATP to help join on that CO2 molecule. This is going to make oxaloacetate in the mitochondria, and this is an important reaction not only for GNG, but also to help the citric acid cycle run. Because remember, oxaloacetate is the final product made and is needed for the first step of the citric acid cycle. In GNG, there's going to be a second step that's going to use the decarboxylation to drive the transfer of a phosphate from GTP onto oxaloacetate. So this is in order to make that unstable phosphoenol pyruvate. The new enzyme that catalyzes this reaction is called phosphoenol carboxykinase, and it's a kinase, so it's transferring a phosphate from GTP onto oxaloacetate. And remember, these reactions would have to run twice so that we have two PEP molecules going through the first half of gluconeogenesis. So as the two PEP molecules go through the reverse sequence of the enzymes in glycolysis, we will end up making one dihydroxyacetone phosphate, or DHAP, and one glyceraldehyde 3-phosphate, or GAP, which will then join to make fructose 1,6-bisphosphate. Step 3 in glycolysis was adding a phosphate on to make this molecule, so GNG is going to hydrolyze it off using an enzyme called fructose 1,6-bisphosphatase. Fructose 6-phosphate will then be isomerized to glucose 6-phosphate, similar to what we saw in glycolysis. The last new enzyme will remove the phosphate to produce glucose, and this enzyme's name is pretty informative. It's glucose 6-phosphatase for the phosphate removal using water, and this enzyme actually plays dual roles. It also works on the glucose 6-phosphate that came from glycogen breakdown, where glycogen phosphorylase made glucose 1-phosphate, and then phosphoglucomutase isomerized it to glucose 6-phosphate. So this is why there isn't a glucose 1-phosphatase for glycogen breakdown. We can just use the enzyme found in gluconeogenesis along with phosphoglucomutase. If we look at the net reaction for the production of glucose from pyruvate, you would need 2 pyruvate molecules along with 4 ATPs, 2 at pyruvate carboxylase and 2 at step 7 in glycolysis that made ATP. 2 GTPs will be required for phosphoenolpyruvate carboxykinase to phosphorylate oxaloacetate, and 2 NADHs will be required at step 6 in glycolysis, which was making NADHs. So for gluconeogenesis, we're going to put in 2 NADHs. There are also 6 waters that will be required in the pathway, all to produce a single glucose molecule. So we do need quite a few energy molecules for these 11 steps of gluconeogenesis, but overall, if there is enough energy in the liver, the pathway will be favorable in order to synthesize glucose. We can also see that glycerol is a great starting molecule to make glucose. It has 3 carbons in it and will be released when we hydrolyze off the fatty acid tails from a triacylglycerol molecule. The fatty acids and their catabolized products, acetyl-CoA, however, can't be used to make glucose, and they also can't be used by the brain to make energy. So instead, 2 glycerol molecules can enter halfway up the GNG pathway as dihydroxyacetone phosphate, along with the use of some ATP to make glucose. So let's take a look at those reactions. Glycerol kinase is first going to phosphorylate glycerol to make glycerol phosphate, and we then see the same redox reaction that we saw when it came to getting electrons from NADH in the cytoplasm into the mitochondria. Only this time we're going to be making dihydroxyacetone phosphate and an NADH. These reactions are going to run twice, and one of the DHAPs will get converted to GAP. The DHAP and GAP can then combine to form fructose 1,6-bisphosphate, and then the 2 phosphatases will remove the phosphates in order to produce glucose. If we compare the 2 pathways, glycolysis is going to be the catabolic pathway that makes energy, while gluconeogenesis is going to require energy, and it does depend on what molecules you start with regards to which energy molecules it's going to need. Glycolysis is going to produce pyruvate or lactate under anaerobic conditions, while GNG is obviously going to be making glucose, or the starting molecule for glycolysis. Glycolysis will be making energy and will essentially be on in most cells as the concentration of blood glucose rises, while in the liver, GNG is going to be able to supply other cells with glucose when your blood glucose drops, either when you're exercising or fasting. Now, in order for a liver cell to know which pathway is going to be on and which one's going to be off, we do need to look at their regulation, and you'll see that the availability of energy is going to be one of the deciding factors. If we look at the last step of glycolysis that had 2 enzyme catalyzed steps in gluconeogenesis, these would be the starting reactions for gluconeogenesis, and they really do depend on the availability of energy. ADP, or low energy, can inhibit these reactions, and this makes sense since they both need ATP and GTP as substrates. We can also see that acetyl-CoA can stimulate gluconeogenesis. It also inhibited step 10 of glycolysis. So if we have lots of acetyl-CoA, say from the breakdown of fats in the liver, then we won't need to break down glucose and can instead share it with the rest of the body. We can also use all that energy coming out of fats in order to make glucose in gluconeogenesis. We can see a similar pattern for step 3 of glycolysis, or PFK1, where fructose 1,6-bisphosphatase hydrolyzes off the phosphate at carbon number 1 in gluconeogenesis. Low energy stimulates glycolysis, where it's also going to inhibit gluconeogenesis. Gluconeogenesis is an anabolic pathway, so if we don't have energy, we don't want it running. There's also this important allosteric regulator that we talked about in the carbohydrate metabolism lecture, so fructose 2,6-bisphosphate. And remember, this was a side reaction that made fructose 2,6-bisphosphate from fructose 6-phosphate when the substrate for PFK1 started to build up. And it can also inhibit gluconeogenesis. Now, we do have to close the loop on this molecule and look at how it's made and broken down, because hormonal signaling can also play a role in the regulation of gluconeogenesis via this molecule. Fructose 2,6-bisphosphate is shown on the right here, where it can allosterically stimulate PFK1 and activate glycolysis, or inhibit fructose 1,6-bisphosphatase in GNG. I told you in the carbohydrates metabolism lecture that it's made by phosphofructokinase 2, which adds the phosphate onto carbon number 2, but this enzyme is actually a tandem enzyme. So I really only told you half of its name. It also has a phosphatase domain. So it's one polypeptide chain that can fold to form a kinase that will add the phosphate, as well as a phosphatase domain that will hydrolyze off the phosphate from carbon number 2. So this enzyme is really important because it also has a regulatory domain, and when this domain is phosphorylated, this will ensure that only one of its enzymatic activities is on at one time. Otherwise, you would have a futile cycle of adding the phosphate on carbon number 2 and then hydrolyzing it off, and it wouldn't work as an allosteric modulator. So let's look at the role of this enzyme. So starting on the left, when we have lots of glucose, glycolysis is going to be running and producing lots of fructose 6-phosphate, and PFK2 is going to be producing this activator of glycolysis. Fructose 2,6-bisphosphate will also be inhibiting gluconeogenesis. So when the kinase is on, the phosphatase domain is going to be off. In order to change its activity, phosphorylation is going to result in a conformational change, inhibiting the kinase domain and activating the phosphatase. This signaling pathway is something that we've seen before. So under low blood glucose, glucagon is going to be initiating the GPCR signaling pathway, where protein kinase A is activated and will phosphorylate this tandem enzyme. This will allow the phosphatase to remove the phosphate from fructose 2,6-bisphosphate so that it's no longer inhibiting gluconeogenesis, and the product, fructose 6-phosphate, can be isomerized to glucose 6-phosphate and then dephosphorylated to release glucose into the bloodstream. Now, what happens if blood glucose rises and we want to change the activity of the tandem enzyme again? There's a phosphatase enzyme that's going to be activated when the levels of fructose 6-phosphate rise, and this can then remove the phosphate from the regulatory domain of the tandem enzyme, reactivating the kinase domain to produce fructose 2,6-bisphosphate and stimulate glycolysis. So this is an amazing example of reciprocal regulation in the liver, where we're centering around the production of this allosteric modulator, And we've got a pretty neat enzyme that's only a single polypeptide chain but has two competing functions. Reciprocal regulation just means that we don't want competing pathways on at the same time. And we also saw this when it came to glycogen breakdown and synthesis. In the liver, hormone signaling can control the phosphorylation of the tandem enzyme in order to produce and break down fructose 2,6-bisphosphate. This molecule, along with a lack of energy, can stimulate glycolysis and inhibit gluconeogenesis, the anabolic pathway. When you have lots of energy, this can slow down glycolysis but will stimulate gluconeogenesis so that it can make glucose for the rest of the body.
So remember, when it comes to energy production, oxygen really is essential. Otherwise, you'd be producing lactate, which could be used in gluconeogenesis. There's also other molecules like glycerol and amino acids that can be used for glucose production in the liver and kidneys. But gluconeogenesis really is an anabolic pathway, so we are going to have to put some energy in, and of course there's going to be 4 different enzymes that are going to bypass those 3 irreversible steps of glycolysis. As always in this course, the regulation of these steps is going to be super important, so do make sure you go over how these pathways are regulated before doing the brain break questions on Quarkus.